Introduction to Scaling Complex Oligos. Scaling complex oligonucleotides isn't just a technical challenge, it's a pivotal step in enabling advanced research and diagnostics. Whether you're working on siRNAs, aptamers, or antisense oligos, the question remains, how do we scale these intricate sequences without compromising quality or purity? Challenges in oligonucleotide scale-up. Complex oligos often feature modified backbones, locked bases, or unusual sequences. These features can disrupt coupling efficiency, increase synthesis cycle time, and lead to product heterogeneity. As scale increases, so does the risk of impurities, requiring precise control over every synthesis parameter. Strategies for efficient oligonucleotide manufacturing. So how do leading chemists overcome these hurdles? It begins with optimized solid-phase synthesis protocols, adjusting coupling time, reagent excess, and solvent purity. Then we scale using high-throughput synthesizers and robust purification workflows, including HPLC and cartridge-based desalting, to maintain consistency batch to batch. Process optimization for complex oligos. For truly complex oligos, like long RNA constructs or those with multiple modifications, scaling means engineering the process itself. Design of experiments, or DOE, helps define optimal synthesis conditions, while inline monitoring ensures synthesis fidelity at every stage. These aren't shortcuts, they're precision-driven solutions that protect both product integrity and yield. BOC Sciences, Custom, Oligonucleotide Solutions. BOC Sciences offers comprehensive solutions for oligonucleotide production, from short primers to long RNA strands, from standard DNA oligos to custom-modified constructs. We provide tailored synthesis services that meet your exact specifications. Our platform also supports cGMP oligonucleotide manufacturing, ensuring quality and scalability for your preclinical or industrial needs. Ready to elevate your project? Contact BOC Sciences and let's make your next oligo a success.